We are Stephan and Samar Sheat from the DSMZ plant virus department in Germany, and we would like to share some of our insights into the development of resistance, virus resistance in cassava. In Africa, we are dealing essentially with two major diseases: cassava mosaic disease caused by cassava mosaic viruses and cassava brown streak disease caused by cassava brown streak virus, two viruses that belong to very diverse families, virus families with diverse biology and causing also quite distinct diseases. Cassava mosaic disease is caused by a number of different virus species, all causing the same disease. It is an endemic disease. It occurs wherever cassava is grown in the continent, and essentially it is spread very, very effectively by the Bemisia tabaci vector. And so we have very limited interference, very limited interventions, with natural resistance being almost the only option for disease management. And thanks to long breeding and selection efforts, immune lines were identified in West African germplasm, and the CMD2, CMD3 type resistance confers immunity, or almost near immunity, against a broad spectrum of viruses, essentially all known cassava mosaic viruses around the world. And this leads us to believe that the CMD is not more a critical threat, at least to the areas where improved cassava varieties are grown. In contrast, cassava brown streak disease is an outbreak, a real outbreak. The disease has been long known in the coastal regions of Tanzania and now spread south and westwards in the continent, and this is mostly by human-assisted spread. We have a very limited role of the vector. This leaves disease intervention options. However, what we also have seen is that the basis for resistance in African germplasm is enormously limited if resistance exists at all. Breeding for cassava brown streak resistance is severely hampered because natural infection conditions are very erratic, and there is a slow disease development, leaving often 8 to 9 months before a symptom can be visible. And more often, unlike what we see here in this picture, symptoms in cassava are not sufficiently informative to identify cassava brown streak viruses. Of course, we have studied brown streak virus since a long time. They have very unique genomes. In particular, this HAM1-like gene is necessary for this virus to infect cassava. So we have a good knowledge about the diversity of cassava brown streak virus in Africa, and this is very important for our selection. And we also know how to handle these type of viruses. We know, for example, that these the different genomes are also representing a different biology. The viruses have completely distinct plant invasion strategies. You see, they cause distinct symptoms, and this is because the viruses unload from the phloem to infect the plants in different veins, mid veins, or smaller veins in the plant. So we know quite a bit about the biology, and before I leave the floor to cassava, I would like to remind us of some definitions that are often used in a bit blurred way. Okay, a virologist, in comparison to a breeder, also looks at the effect on the virus itself. So when we assess plant responses, we just say, yeah, it has symptoms, or it has no symptoms, and this is sensitive or tolerant. But we also say, what is the effect on the virus by itself? And I think this needs to be taken into consideration, because what often is called tolerant is still a plant that is infected with the virus, and in particular areas can these tolerant plants also show very, very strong symptoms. So I need to remind you, we look at these virus resistance or virus response in the plant in a different way, and infection, susceptible, resistant versus sensitive and tolerant need to be properly addressed if we all want to have a harmonized way forward. So I now leave the floor to Samar in order to tell you about our approaches to look for cassava brown stick virus resistance.
So I would like to share with you our experience searching for cassava brown stick resistance. So for that, first we have to identify where we have to search for cassava, and we have the tool for that, identify the resistance, and then to test this resistance and durability of the resistance under field condition, and in later stage to see why this plant is resistant. First, to screen for resistance, you have to have a tool, and this tool is first how to propagate cassava and try to maintain cassava. So we know how to deal with the cassava in tissue culture and also on seed, how to germinate seed. And the most important things to know which age is important to start the screening. And in our case, we decided to work with 2 months cassava plants. The next was the virus infection method. So as you already know that the infection, it's happened naturally by Bemisia tabaci. However, also in our laboratory, we can do this by agroinfiltration or by bombarding and by injected also by body grafting. We decided to do the grafting using the body grafting or side grafting because in this case, we are sure that the plant is infected because we use a bud from systemic infected leaf or an infected stem. And also we know the time of the infection. So we guarantee that all the plants are infected on the same time. And the most important thing is that all plants are infected. And we increase our efficiency by two body grafting. In this case, we guarantee that you have 100% infection in your plant. So the third thing was to which isolate we have to use to start the screening. As Stephan already said that we have a big diversity between cassava brown stick isolate and species. The most severe one is CBSD Mozambique, as you see here in the Pantamiana plant, and also in the cassava plant, Albert and Namikonga, that we know that they are totally different in their response to cassava brown streak virus. And we see that CBSD Mozambique is the most virulent virus in terms of the severity of the symptoms as well from the virus titer through the plant, because it has a high virus titer in the Albert, which is considered highly susceptible, and Namikonga, which is considered a resistant African plant. And also CBSD Mozambique has a first onset of the symptoms. So we decided to use first the CBSD Mozambique to eliminate as much susceptible plant as possible in the first round of screening. The next was to see which tool we have to use for virus detection and quantification. As you already know that we have quite a good protocol, ELISA protocol, to detect the virus titer and quantify as well. And as you see here, it's quite reliable because you see the difference between the healthy and the infected plant, also the different isolate. As well, this is good only when you have a highly susceptible plant. And if you were screening for resistant, and if you want to be sure that you are catching everything in the virus, then you come here and see the difference between the viruses, between the RT-qPCR and the ELISA. So when you have a plant infected with a CBSV or UCBSV, and you have a symptom score 1, and the ELISA is negative, you see that RT-qPCR is still positive. And that proves the highly sensitive method is really important when you do a screening, and it's also important when you do quantification. So we also want to know from which part of the plant we have to sample, and as we know already that the virus titer is differ in the cassava leaves, and it's concentrated in the lower part. Not only is different in the cassava leaf, also different by time. That you see here in the susceptible plant, the infection start earlier, while in the resistant plant, Namikonga, the infection starts so late. So you have really to have the timing right to screen for resistance, because this is important when the infection starts. So 4 months between 4 to 6 months is it's good enough to also include the resistant one. Also, we know that cassava has also the stem and tuber, and it's play a major role in the infection. And as you see here, also tuber has accumulated a high virus titer. So we should also don't forget the role of the tuber in the infection. And this is not only in susceptible plant, Moreover, also in the resistant plant, when you have here a Namikonga and Nachinyaya, you see when you cannot detect in the leaf, you still can detect the virus in the stem and the root. So our protocol was considered taking a sample from leaf and stem and tuber to be sure that we screen the whole plant. Also, on the resistant cassava plant, which has, for example, in the root system, which is consistent between 9 to 11 root tubers, when one tuber shows symptoms and this symptom scores for 5, for us is considered not resistant. Because when the plant is able to replicate a virus and show a score 5 symptoms, even in one root, this is considered for not resistant. So we have also to take this in account when you screen for resistant. And this also points to the role of the root in the cassava brown streak infection, and also point to the cassava brown streak as a root virus because of the high virus titer that we find already in the root and also in the resistant plant. So when we screened using the South American cassava germ plant, we could see that the cassava responds differently to CBSD infection, and from the plant side, we see a plant that highly sensitive type develop a dye pack shortly after the infection, 10 to 15 days, or highly tolerant one that didn't develop any symptoms at all. But we were interested more to see if this line, the virus still is present in this tissue. So we screened the leaf and the stem and the tuber as well, and you see we were lucky to find a group of plants that the virus was not there, so they were virus-free. And this we called it immune. And also we found another group of plants which restrict the virus to the root, and we call it resistant by root restricted because the virus was visible in the root and also by necrosis. It developed necrosis. So we asked ourselves why the South American cassava is resistant and based on what reasons. So as you see here, using in situ hybridization technique to localize the CBSV in a cassava leaf, from different cultivar, African cultivar, and also using our South American resistant cassava. The red dots here represent the virus titer, and the more dot cluster also represent the more virus, meaning more virus replication. And you see here also in the susceptible and resistant plant, virus replication was present, despite that there is a difference between them. However, in the South American cassava, there's no virus at all, and only rarely was found, and this only because we put the virus inside the plant. When we look also for the localization of the virus in this case, we see also the virus in the resistant cassava plant, the South American, was only restricted to the phloem, and in this case here only to the external phloem in the immune line, while in the root restricted one was to the external and internal phloem as well. So this was in comparison with a highly susceptible plant where the virus was able to invade and replicate and mostly all kinds of cells. So we proved that the virus is restricted to the phloem cell, and we also were able to define the phloem cell that the virus was using, which was a companion cell. In this case, we summarize that the resistance in our South American cassava is based on restriction in virus replication and virus movement. As we now know, the South American cassava and why this plant is resistant, the next was to use this resistance in the South American cassava in the African breeding to breed for dual resistant for CBSD and CMD, because unfortunately the South American cassava were highly susceptible for CMD. So it was really good that if we can use this plant to breed for a dual resistant and also to develop a high throughput virus screening protocol. So this already done because our partners here at TARI and ITA were crossed this South American resistant with a CMD resistant cassava, and we were able to receive different population. And from the first population that we get from CR, from Xiaopei, we see that they were able to find the resistant in the F1, and this proved that cassava brown streak is a dominant trait. Actually, to reach this, we have to develop our password virus screening workflow. In this case, to screen for 2 virus or two disease at one go in a short time. So in the first round, in the phase 1, as you see here, we screen for CBSV, and all the candidates, the negative one, then enters the UCBSV ACMV screening in the phase 2, and then all the candidates can reach where negative in the first confirmation cycle goes to the field or to the second resistance screening. And based on our protocol, as you see from the from the timeline, in 9 months, we were able to reach the field. So in 9 months from seed, we were able to reach candidate, which we brought to the field. This compared to the breeding protocol that normally, classical breeding protocol that normally takes around 5 years. So in our case, we reached the field in 9 months. Here, we make a shortcut where we can reach the advanced trail the year 5 in the breeding protocol. Actually, what we see that cassava resistant against CMD and CBSD is real, and we have already our first candidate in the field, and we hope that this is a solution for CBSD and CMD disease. For that, I would like to thank you, and we